webinar, which we hope to be a long-standing and a long-running webinar series. I want to first start by saying a few words about our society. We were formed in 1903, and as such, we are one of the oldest scientific societies in the United States. Our major goal is to bring basic scientists and clinical scientists together in an effort to facilitate exchange that brings and accelerates scientific discovery to the practice of medicine. I want to play a very short video of an interview between of myself and another past president, Dr. Mike Friedlander, as we attempt to answer the question, what is SEBM?
I think the importance of the Society for Experimental Biology and Medicine is captured in its name. That is the very confluence of experimental biology and medicine. There's never been a time in history where the interaction of these two disciplines has been more important for people, for the health of our nation, and the health of the world. It really brings together scientists and clinicians in a meaningful relationship.
We were founded by clinician scientists who were interested in understanding the basis of what it is they're doing in terms of therapies. And so one of the benefits that we have is being able to translate that to be really bench to bedside and help with driving those discoveries. And because of the interdisciplinary nature of our society, to have the capacity to translate that into newer modes of healthcare and better healthcare.
One of the things about the Society for Experimental Biology and Medicine that differentiates it is it has brought together and kept together people from multiple disciplines. So the whole buzzword of interdisciplinary and multidisciplinary research is the very ethos of this society. It lives it and has every day for the last century. So there's never been a need greater than now to have this kind of truly interdisciplinary organization. So SEBM is really in the lead in this area.
So as discussed, we emphasize the translation of scientific discovery to hopefully improve healthcare. Another reason that I have personally continued my affiliation with SEBM is our strong commitment towards the mentoring of young scientists. We firmly believe that they are the future of science and medicine, as we were once in our day. In fact, Dr. Emma Bowers, one of a postdoc that's in the society, will be moderating today's session. So if you're a trainee or a young scientist and you're looking for a group of folks that is vested and has a vested interest in your career development, I'd suggest that you check out our programs and so forth for the society. SEBM is also affiliated with a journal. To share again. Oh, there we go. The experimental biology, called Experimental Biology and Medicine. As scientists, we all need a forum that we can publish our discoveries, and this offers this forum. I wish to just in general speak, it listed on here as a variety of attributes for this for the journal. I wish to just point out a couple of these. It is strongly supportive of our interdisciplinary aspects of the SEBM experience. It has a global audience, and it's currently has an impact factor of 3 and rising. Dr. Steve Goodman, the editor-in-chief of EBM, will be leading next week's webinar discussion, and will fill in some of the details about the journal. Turning to today's topic, when we began discussing the topic, it was clear to us that we were approaching it as a scientist does to a new issue that they wish to study. That is to say, we wanted to begin by getting an understanding and know everything that is known about the virus, about the mechanisms, and its both present and its historical concept. Secondly, we wanted to understand what tools are presently available that can be brought to bear for the issue. And finally, and thirdly, we wanted to, what other avenues might we explore or to pursue to attack the issue or attack the virus. Today, we start a series of 3 webinars concerning the COVID-19 virus by going through a background and history of basic research of the virus. We have 2 internationally renowned speakers for today, Dr. Stanley Perlman from the University of Iowa and Dr. Kyle Harper from the University of Oklahoma. So by way of introduction, let me introduce Dr. Stanley Perlman. Dr. Perlman is a professor in the departments of microbiology and immunology and pediatrics at the University of Iowa School of Medicine. He holds an MD from the University of Miami School of Medicine and a PhD from Massachusetts Institute of Technology. His research long focuses on the pathogenesis of coronavirus infections. His talk today will discuss the SARS-CoV-2 basic research in new issues. Dr. Perlman, the floor is yours, sir.
Thank you. So what I'm going to talk about today as the introductory to this series of webinars is to describe coronavirus structure, replication, and pathogenesis, and a little bit of the history of where we are in terms of what we know about coronaviruses. And much was known before the COVID-19 pandemic began, but I have to say there's been many surprises that this virus has shown us. So I have no conflicts of interest. So our learning objectives in this webinar are to learn a little bit about the structure of coronaviruses, how do coronaviruses enter cells, what is the replication strategy used by coronaviruses, and what is the pathogenesis of coronavirus infections. And I'm not going to be able to cover all of this in great detail in the time allotted, but I'll try to do the best that I can. So first, starting with the structure of a coronavirus, coronaviruses were first really discovered by the biomedical community in the 1940s and 50s, and they were isolated from farm animals that were infected with these viruses that caused diarrhea and respiratory disease. And they were initially, of course, like all of the viruses at that time, not well characterized until electron microscopy was done, and I will show a picture of that in a little bit. And because of this structure on the outside of the virus, this was labeled a coronavirus because, as I'll show in the EM picture, it has the resemblance of either the corona of the sun or the corona of a crown. And so that's how it's got its name. These are single-stranded RNA viruses, and they're very large. So coronaviruses are 31 kb in size. They're positive sense, which means they're RNA viruses that can be directly translated. The RNA can be directly translated in the cell to make proteins. And these are members of the nidovirus family. And this is not the biggest member of the nidovirus family. For the longest time, we thought that 31 kb size of the coronavirus made it the biggest virus. But in fact, there's related viruses in the nidoviruses that infect insects that are 40 kb. So these are even bigger. And this particular family of viruses are the coronavirus. And as I'll discuss in the first few slides, before the human coronavirus epidemics and outbreaks and pandemic began, we mostly knew about these viruses because they infected cows, they caused a common cold in people, they caused the diarrhea in farm animals, and many of these are well known, especially in my state, Iowa, transmissible gastrointestinal and transmissible gastrointestinal virus, porcine epidemic diarrhea virus, and porcine delta coronavirus all cause diarrhea in baby pigs. And then the these viruses can also cause brain infections. We've studied for many years mouse hepatitis virus. There's a virus that causes brain infection in swine and in rats. And the neurotropic ability of this virus, of course, has become more compelling as we've learned more about COVID-19. So these are, as I mentioned already, they're broadly diverse across mammalian and avian species. And I didn't even mention that some of these viruses infect mostly chickens, and the same viruses that infect chickens also infect beluga whales. I'm not sure why that's true, but it is. And for human coronaviruses, before the COVID-19 epidemic and before the SARS epidemic, we knew about two human coronaviruses, OC43 and 229E. And as we've learned more about COVID-19, it turns out our information about these viruses is actually useful to our understanding, because these viruses cause upper respiratory tract disease, and COVID-19 is both an upper respiratory tract disease in the majority of people and a lower respiratory tract infection in a few. And these are the people we worry about, of course, because they get the severe pneumonia, and they are the people who die from the infection. After SARS was discovered in 2003, two new human coronaviruses, NL63 and HKU1, were also identified. And these have very similar disease manifestations to OC43 and 229E. They basically cause the common cold. And I've already mentioned the recent zoonotic coronaviruses that had potential to spread around the world, SARS-CoV and MERS-CoV. And of course, SARS-CoV hasn't spread around the world, but its related cousin, SARS-CoV-2, that causes COVID-19 certainly has. Nidoviruses are classified as I show here. The nidovirus is the order, and then the family is coronaviruses. There are 4 types of coronavirus groups, the alpha, beta, gamma, and delta coronaviruses. And then there are these toroviruses, which affect animals. We've never seen any of them infect humans. They're very similar in size to the coronaviruses, but they're less well studied. And then there is other related viruses in the nidovirus family. So this is the structure that I was describing a little while ago. You can see in the electron micrograph, you can see these spikes, and this is what gives it its name, the coronavirus. And the virus itself, on the right side, you can see this nucleocapsid RNA protein complex, which fits inside this shell of proteins. The spike protein, the one we can see in the EM, is the most important for binding for cells, and is the one that people hear the most about because it's not only important for binding to cells, but it's also the target for the antibody and T cell responses. There's other proteins shown here in black, the M protein, and these little red ones, the envelope protein. And these are all required to make a shell around the virus. So before 2002, coronavirus research focused on one of the viruses I cared about, murine coronavirus. This was isolated in 1949. It was actually from a lab at Harvard University where an investigator noticed a mouse with hindlimb paralysis. And I've studied this virus for many years because in many ways it has similarities to the disease, the human disease, multiple sclerosis. And so this has been interesting. And what we've learned from this virus, which was the most widely studied virus until 2003, we've learned many things that have been useful for our understanding of COVID-19 and the SARS and MERS coronaviruses. And then I already mentioned this a little bit. The other thing I should mention is the animal viruses, and I'll mention this again. The animal viruses actually gave us information about how you make a vaccine against coronaviruses, because especially the viruses against, well, all of these against pigs, cows, cats, and birds all cause economic loss. And in the case of cats, caused, it's a devastating, the virus we care about is a devastating disease of cats. So we became interested, the research community became interested in vaccine development. So we learned a lot about how vaccines should and shouldn't be developed when we look at these animals' pathogens. This just describes, again, some of the viruses that we've known about in the past. These are the organs that were infected. We know the lungs were infected primarily by actually avian infectious bronchitis virus and a bovine coronavirus before the SARS and MERS epidemics. And then we have these others listed here, which I discussed a little bit already. And so coronaviruses, as we've known for many years, are global zoonotic threats. We know that before COVID-19 was recognized as a disease, we knew that the SARS coronavirus crossed species in 2003 probably from bats and then on to exotic animals like Himalayan palm civet cats. And then other viruses like the MERS virus is endemic in camels. There's a lot of really interesting questions about MERS virus, which I'm not going to go into in great detail, but MERS coronavirus infects camels all around the world, but only does it transmit to people to any significant extent in Saudi Arabia. And even though it's been in Saudi Arabia for many years, it didn't do this to 2012. So there's questions about why is this happening that are generally scientifically interesting. Of the human cold coronaviruses, 229E and NL63 came from a common ancestor about 1,000 years ago. NL63 uses the same receptor to enter cells as does the SARS-CoV-2, the agent of COVID-19. And these viruses entered populations and now cause the common cold. Some people wonder whether 229E and NL63 didn't cause a major pandemic like we're seeing now, and then changed into a cold coronavirus. Of course, that's what people hope that COVID-19 virus will do. It'll turn into a mild cold rather than being the severe pulmonary infection that it is in some people. So, and here's another example of a virus crossing species. This was described in Nature 2 years ago, but this is a bat virus that directly crossed species to infect pigs and caused a fatal diarrhea syndrome. And so the people who did this, these are actually the people at the Institute of Virology in Wuhan. And what they discovered is the virus was nearly identical to a bat virus that they found in caves in China. Because this group has gone out and tested lots of different viruses. They examined lots of different bat viruses and found the ones that seem to be the precursors for SARS-CoV-2. And in this example, for whatever reason, bats in China seem to be more likely to cross species to infect humans. There are plenty of bats in the United States. Whether they will do this or not is really an open question. And this is another example of how viruses cross species. This virus, bovine coronavirus, probably began in cows, crossed species to become the human cold coronavirus OC43, and then crossed species again to infect alpacas and deer and many other ruminants. So this is a disease that readily spreads across species. And this is a theme about coronaviruses. They like to cross species. So this slide, I hope it's clear. It looks a little blurry on my screen, but so coronaviruses enter cells in two ways. And particularly for the lung coronaviruses, we have a little more information about what's most important. So in one scenario, the virus enters the cells at the surface. And this matters because what it does is it enters by binding to its receptor, which in the case of SARS-CoV-2 is ACE2. And then it requires cleavage by this enzyme called TMPRSS2. So there should be a 2 at the end of this. And then after that, it enters the virus, the genomic material directly enters the cytoplasm. In a second way of entry, the virus can go into endosomes and then go and fuse with the cellular membrane in these endosomes and with the involvement of cathepsin. And this matters because in the discussions that are very common in the press now, there's a drug called chloroquine or hydroxychloroquine, which inhibits this step, this cathepsin step. But in the lung, this is the minority of the entry pathway. Most of the virus enters via this TMPRSS2, ACE2 and TMPRSS2 pathway rather than an ACE2 cathepsin pathway. So this is the reason that many of us were skeptical that chloroquine would actually, or hydroxychloroquine would actually work, because it affects the route of entry that's least used by this particular set of coronaviruses, the human respiratory coronaviruses. And so the other thing that's shown here is this S1, S2 cleavage. And this is another site that's been discussed because it has the resemblance of a furin site, which means that it's readily cleaved by many enzymes, including furins, which are very commonly in cells. And this may contribute to its pathogenicity, because when you have a furin cleavage site, the virus can be cleaved as it leaves a cell rather than just on entering a new cell. This may give it a little chance to enter a cell more quickly and therefore not be detected by the immune system. This is not known for sure. There are furin sites in other coronaviruses as well, but not in SARS or MERS. And so this is again a discussion of the SARS, showing more schematically binding to the spike, to the ACE2 receptor, and then cleavage by the TMPRSS2 protease. And then this is a complex, this complex of the receptor and TMPRSS2 and the tetraspanins are all, has been well defined and is important for surface entry. And this is actually a useful target, I think, if one wants to prevent entry into cells in the lungs. And there's drugs like camostat and namostat, I think it's its name, that seem to inhibit TMPRSS2 and may be useful. But they're only approved for use in Japan and actually for pancreatic disease. So this I'm not going to spend any more time on, except to again show this, the basic pattern of entry is you have an S1, S2 cleavage site, and what's key afterwards is to have this S2 cleavage site shown, S2 prime cleavage site, which releases a fusion peptide, which is important for cellular entry of the virus. And this shows a picture of the spike protein of MERS coronavirus. In this case is the MERS coronavirus. We of course now have structures of the COVID-19 virus S protein as well. And they're all very similar. And what's very remarkable about these viruses is that they all seem to like to bind to these large ectopeptidases, which are proteins that cleave other proteins. And so both the SARS, which binds to ACE2, and MERS-CoV, which binds to dipeptopeptidase 4, and even MHV, which binds to CCAM1, which is an adhesion protein in the gut and elsewhere, and also on T cells, other cells as well. For some reason, these viruses really like to bind to these proteins, but they don't use the protein function, because you can remove all the protease activity from these target molecules and they still work just fine as receptors for the virus. So I think I will, I just want to make one point here. So particularly for SARS-CoV and MERS-CoV, and much less so for SARS-CoV-2, we know from the cryo-EM work that's been done recently, because cryo-electron microscopy allows you to see large molecules. We know that the structure of the part of the molecule that binds to the receptor is either in an up or a down state. And when it's up, it can bind the receptor. And when it's down, it can't. So the cryo-EM allows you to see these two states. So we've really learned a lot. And this was before SARS-CoV-2 was even identified. So we learned a lot about how these proteins might work in binding. And we care because, as I said earlier, not only is it important for binding, but these cells are also a major target for the immune response, both the antibody and the T cell response. So we want to know as much as we can about them. And so in thinking about ACE2 expression, ACE2 is expressed mostly on one kind of cell in the lungs, a ciliated cell, and MERS coronavirus uses DPP4, which is expressed on cells that are in the alveoli and also in Clara cells, which are the non-ciliated cells in the lungs, and also include the goblet cells, which make mucus. So you have these two differences. What's really remarkable about COVID-19, I don't have a picture here, but the SARS-CoV-2 seems to really cause destruction of this layer, even in the test tube. These other two viruses both infect cells in the test tube, but they don't cause that much destruction of the cells. But the SARS-CoV-2 seems to be more destructive for whatever reason. And so the other thing I should say is that the SARS-CoV-2 receptor, the S protein, binds to its receptor better than the SARS-CoV molecule did. So it's really well adapted to infecting humans. So after the virus enters cells, what happens, you can see on the left-hand side, is that you have the virus is uncoded, as I mentioned, and it's translated into proteins. And the proteins are these large proteins that are then chopped up into pieces that I'll go over quickly in the next slides. And after that, the virus ends up reproducing both by transcription of RNAs and replication of the entire genomic RNA. And then after that, the virus is formed in these areas in the cell next to the endoplasmic reticulum and Golgi called the urgic. And from there, the virus is released into vesicles and they bud out into the fluid around the cells or into the body. So the replicase proteins are why these proteins, the ones involved in making RNA, are why they're so big. These encompass 2/3 of the genome, and we don't know why so much information is needed. Certainly these viruses seem to do a great job crossing species. So we wonder if this doesn't contribute to it. These proteins are really, it's not like that we know everything about them, but they've been well characterized because these proteins are the same, or basically the same in all coronaviruses, ranging from mouse hepatitis virus to chicken viruses to the SARS-CoV-2. And because of time issues, I'm not going to go over all this in great detail here, but I'll just show a couple of points. So the replicases, as I mentioned, is a huge RNA protein. It's translated, huge RNA, and it's translated into several proteins. Most of the protein is this what are called the non-structural proteins 1 to 10. But then this is a sequence here that allows the ribosome to slip, and it makes these other proteins, which are 12 to 16. And these proteins are actually the replication proteins, and they're made about 1/4 as much as the ones at the other end, at the 5' end of the RNA, or the N-terminal part of this protein. And we think the reason for that is so we can control the amount of protein that's actually involved in replication. One of the proteins that's really important is the RNA-dependent RNA polymerase, and this is the target for remdesivir, which is which may be a useful therapy in patients who have COVID-19, assuming that it's delivered soon enough, early enough in the infection. And I mentioned already the 16 proteins. I'll skip this. And so transcription begins at the protein is made, or the 5' end of the protein makes, the RNA encodes the RNA-dependent polymerase, as I mentioned. That then hops to the 3' end of the RNA to make a negative strand, because since this is a positive strand RNA, it enters into the cells directly and is directly translated. And after that, then you make a full-length negative strand RNA, and at the same time, you also make these smaller RNAs. And these RNAs matter. They're negative strands. They're made by this polymerase stopping at these TRSs and then hopping over to pick up this leader sequence way at the other end of the genome. And these proteins are then translated into positive strands, which are then used to make the proteins that are important for the virus. And for those proteins I showed you before, either the 16 non-structural proteins or the nucleocapsid and spike proteins and membrane and envelope proteins. And then once the proteins form, NSP3, 4, and 6 make these structures for which replication occurs. And what the virus does is it manipulates the membranes of the cell. And these structures then end up looking something like this, these double membrane vesicles, where we think RNA is inside here. And RNA is also found outside here. So the virus it uses all these membranes to develop strategies both for replication. And if you have your RNA in the middle of something that looks like this, it's very hard for the immune system to see it. So that's another advantage of these convoluted membranes and double membrane vesicles. And this is what they look like under the microscope. You can see the nice double membrane here around a vesicle. And this is here, you can see convoluted membranes. And then the final thing in the genome structure are these accessory proteins, which are important for probably for invading the immune response. These are the ones found in SARS-CoV-2. What's really curious is you can delete a lot of these proteins. We know that best for MERS without seeming to change pathogenicity. So it's all not straightforward exactly how these proteins function. I don't know if they're mostly important in bats or some other species, but they're not super critical for the human infection. And we know both in SARS-CoV and SARS-CoV-2 that part of this protein, the 8 protein, changes and it doesn't seem to affect anything that we know about yet. So with that, I will stop and turn this over to Kyle.
There we go. Can I jump in? All right. Are you all seeing my screen here?
I don't think we have it.
No.
We did, and then
We did.
And then I think maybe it was minimized or something.
Give me hit share screen again.
Yeah.
Try this. I don't know why that's. Are you seeing a house picture there?
No, I saw the slides.
You saw the slides.
But now I don't.
I don't. Okay, let me try this again. Sorry about this.
No worries.
Do you have it now?
Yep, you got it.
Okay. Can I jump in?
Yep, go for it.
All right, great. Well, thanks very much for inviting me to participate in this webinar. I want to confess that when I was first invited, I Googled Kyle Harper virologist to see if you meant to invite somebody else, and was delighted when it apparently turned out that it wasn't a mistake, or you decided just to ride with it. But I'm really a historian, and I have worked on the history of infectious disease, and we're all living through history, and you all are mostly living through it as microbiologists. And I'm living through it as a historian who's worked on and thought about the impact of health and disease on human history, and particularly pandemic events, and most recently on pandemic events in the Roman Empire, which was struck by really large-scale pandemics that may have been caused by everything from smallpox to the bubonic plague. But more generally, think about and work on the way that human history shapes ecology, and the way that shapes pathogen evolution. And it's from that perspective that I approach the question of what do we know about the history of coronaviruses, what don't we know, and what might we be able to know. And as Stanley showed us, there are now 7 species of human or coronaviruses that are known to infect humans. And most of these are relatively recent. Of course, SARS-CoV-2, SARS coronavirus 1, MERS, that's emerged sometime in the last few decades. And then these other species that he mentioned, HKU1, which is thought to have emerged in the 1950s. And then a few older species, OC43, which is pretty well dated to the 1890s by molecular clog methods. And then these 2 other species that he mentioned as well, 229E and NL63, which seem to be older, although those estimates are much more uncertain and very sensitive to whether or not we have the most closely related species or lineages. So those dates are by far the most uncertain estimates, but they're still very interesting. And they kind of provide us a starting point to ask, why do we have so many coronaviruses, and what can we know about the history of this family and its relationship with human health, and how do we know it? So I'm going to start by zooming out in the very big picture and think about the even deeper question of why do humans have so many respiratory viruses? If you look at our closest relative, the chimpanzee, it not only has far fewer species of pathogens, but it has a different sort of mix in its portfolio that's much more dominated by helminths and protozoa. So our closest primate relatives have worms, and they have sort of malarial diseases. And it's even somewhat misleading because most of the viruses that have been detected in primates, particularly chimpanzees, although certainly not all of them, are actually human viruses like respiratory syncytial virus that they've somehow picked up because of us, because we got primates that were under surveillance sick. So humans really are strange in being primates that have such a heavy burden of disease from bacteria and viruses, and particularly from respiratory viruses. And to ask why that's so is really an ecological question. And in very broad terms, the answers are because we're an urban animal. We live in huge settlements. Other primates live in relatively smallish groups and have relatively small overall populations, whereas there are 7 billion of us. And moreover, we've surrounded ourselves with domesticates like cattle and camels that play an important role in coronavirus ecology, as well as commensals that in many other cases are important, either reservoirs or vectors of diseases. So if you look at the world through the perspective of a coronavirus, you look at humanity and you see a lot of lungs that are really close together that you can infect. So one way to think of it is that we're sort of primates that live like bats, that is in very large, dense populations that are a pretty attractive host from a virus's perspective. How can we know about the history of a family of viruses like coronaviruses? It's actually very hard as a historian to think about how we might come at this from human sources. Human written sources about the history of disease are very limited and very tricky. Part of the problem is that there's very limited, or in most cases, no surveillance of infectious diseases before the 20th century. And there are important exceptions like London, where there are records of cause of death that go back to the 17th century. But at least before the mid to late 19th century, the disease categories that they use don't map easily onto what we recognize as infectious diseases. So they don't have germ theory. They don't think of diseases as necessarily specific phenomena, and things like fever are considered a disease rather than a symptom. So in most cases, it's very difficult to offer a specific diagnosis from historical records. Sometimes you can in a disease like bubonic plague, which has very distinctive pathology as well as epidemiology. So if half of the country dies in the space of a year and there are giant swellings under the armpits, that's probably a dead-on ringer for bubonic plague. But respiratory diseases, as well as gastroenteric infections, have very indistinct clinical presentations. So most of our historical accounts of illness and death aren't distinctive enough to identify that was an influenza or that was a coronavirus, and it's a problem I'll come back to in a minute. So where historical sources start to fail us, we can look to genomes as a kind of historical archive that have their own challenges and limits, but also tell us things about the human past and our relationship with disease and health that we didn't know. And as a historian, I think it's a very exciting time to be alive with high throughput sequencing that's rapid and affordable. The amount that we're learning about the history of our infectious diseases simply from contemporary genomes and modern phylogenies is really extraordinary, and it's helped resolve some of the oldest and most challenging questions about the history of infectious disease, and it's contained many surprises. Phylogenies have told us that we didn't get tuberculosis from the cows, we gave it to the cows, so sometimes it's totally changed our knowledge of evolutionary relationships. Sometimes the phylogeography of an important pathogen has changed the way we think about its history. So historians used to debate whether malaria existed in the New World before the crossing of the Atlantic, and that's been definitively resolved in the negative. It was brought across the Atlantic by European and Africans who were migrating. It's helped us follow the adaptation of very specific and important pathogens like bubonic plague, where it's now possible to identify the acquisition of very critical virulence factors, in this case plasmids, that made it one of the most lethal diseases in human history. And it's possible to time evolutionary events. And this is particularly interesting in the case of a virus that we'll talk about a little bit more, the measles virus, another respiratory virus, whose deep history is being created in a way that we couldn't have imagined even a few years ago. Here you see a phylogeny of morbilliviruses that tell us that the measles virus sits on a branch that's surrounded by other animal viruses, and then more broadly by a huge number of rodent viruses. And so we can infer from that pieces of a story of a history that we simply had no idea about before. I think the overall big picture of the history of coronaviruses is that human progress has accelerated the emergence of diseases. And so here you see plotted the number of humans on the planet over the last 1,000 years, and the inferred emergence dates of the 7 species of coronaviruses. And you see that the evolution and persistence of coronaviruses in human populations is a result of the obvious expansion of our species in sheer numbers. But it's also the way we live. And I only spend just a few minutes on this slide. This is forthcoming work that will appear that is, I think, a very revealing study of another respiratory virus, in this case, the measles virus, that shows the way that I think molecular clocks and phylogenies can help us understand how human ecologies have promoted the emergence of important diseases. Measles virus is in many ways a virus that shouldn't exist. It's extremely contagious and confers very stable immunity on survivors. And we know that it moves through populations extremely quickly. And one consequence of that is that it takes large populations to persist. It's a human specialist. And so there aren't known animal reservoirs. So it needs the ability to move continuously from human host to human host. To survive, to avoid extinction, measles virus requires a population of human bodies of 250,000 or larger, or it will go very, very quickly extinct. So we knew that measles virus wouldn't have survived without the rise of cities. But this is forthcoming work from a remarkable team at the Koch Institute that managed to sequence one of the oldest RNA viruses ever sequenced from 1911 from a formalin-fixed lung tissue of a victim of the measles virus. And that helped to calibrate the molecular clock, and also they used selection-aware molecular clock estimates to revise our understanding of the emergence of measles. And it dates the emergence of measles, you see in the red, the probability distribution, to around 2500 BC, and that's what the top chart shows, that measles virus diverged from its closest relative, the rinderpest virus, which is a bovine morbillivirus, around 2500 years ago. That's the top diagram. And you see plotted on the same time scale, the bottom diagram is a chart where each dot represents the largest urban settlement in 3 different regions of the world. And so you can see, I think it's remarkable, the dashed line is the critical population threshold for measles. So it's 250,000. Every dot below that is a city smaller than 250,000, every dot above that. So what you're looking at is the rise of urban settlements, urban ecologies. And I think what this story tells is that the measles virus emerged almost simultaneously with the rise of human cities that were big enough to sustain it. So it's a neat story of how viral emergence has gone hand in hand with human created ecologies. And measles virus, I think, is revealing too, because it really is a virus that would have gone extinct if it had infected any other primate, or if it infected humans 1,000 years earlier. I think one of the probably unrecoverable features of the history of coronaviruses is there probably were coronaviruses that transiently infected human populations at earlier stages of our history that haven't managed to either avoid extinction or to become permanently established. So when we think about the 6 or 7 coronaviruses that infect humans, we're looking at a subset of those that that have survived or established in human populations, of course, with the exception of SARS coronavirus 1, which probably would have managed to establish itself if it hadn't been subject to severe public health interventions that were in that case very successful. So coronaviruses, I think, also belong to the subset of respiratory pathogens that have evolved in response to the opportunities that human societies have created. I think it's very hard to recover their history from the written record, but it's worth noting that there is a pattern of planetary waves of acute respiratory disease over the last 500 years or so, in waves that come on 10 to 40-year periods, and that have usually been thought to be influenza. So people have normally looked at these and thought these are influenza, and for the most part, they probably are. These very fast-moving, widespread acute respiratory pandemics. But only from 1918 onward is it definite that these are influenza pandemics, because from 1918 onward, we have genetic identification. Prior to that, these are inferred to be influenza, but that's simply from the symptoms that are described in the sources. And those obviously could be representative of emergence events of the coronavirus. And in one particular case, I think there's at least some argument for one of these waves being the coronavirus, and that would be OC43, which, as Stanley mentioned, is a ubiquitous pathogen. It's a common agent of the common cold. It's very closely related to a bovine coronavirus, and Vigen et al. Have used molecular clock dating to estimate its divergence from a bovine coronavirus about 130 years ago. Humans love beef. I know that well, being here in Oklahoma. And one of the features of the world that we've created is that cows are a very successful species and represent an important source, reservoir, and bridge or intermediate host for the emergence of new pathogens. This virus, OC43, emerged right around 1890, and that's a pretty sound date, probably within a few years, a pretty good estimate. But again, this is only on molecular clock methods. There's no RNA that's been sequenced, although in theory that's possible. Somewhere out there probably are tissues from victims, and increasingly bioarchaeologists are able to extract proteins from dental calculus that might be able to identify the agent of some of these past pandemics, including the supposed Russian influenza of 1889 to 1892. And so Vigen et al. Even raised the possibility that maybe this supposed influenza that went global within months, that recurred for 3 years in 3 successive waves that brought severe respiratory disease, that progressed to pneumonia, that had extraordinarily high morbidity rates, so 40, 50% in many countries in Europe where there was reasonable data. Fairly low mortality, except for the elderly. So it'd be consistent with a coronavirus, but obviously also potentially with an influenza. So it might be possible using genetic evidence and historical evidence to trace the emergence of some of the earlier coronaviruses. So I'll close there, but just say that I appreciate the chance to share in this venue. I think it's an example of consilience. I know that biology enriches history. I say that as a historian. I hope that history can at times enrich biology by helping us think about the ecological context for the evolution of our pathogens and understand their emergence more deeply. Maybe even somewhere down the road remotely influence our thinking. When we treat a human being clinically, we're really treating a primate who's exposed to a really extraordinary disease pool that is a product of our unique history. So thanks for inviting me. And if you're ever looking for a historian, there's my email address. Don't hesitate to reach out.
Thank both of the speakers. Kyle, not an interloper. I read your recent Time article, and it was fascinating. And I thank you for a fascinating topic and discussion. I want to turn it over now to Emma Bowers, who has collected.
Hello, can Can you
so
hear
forth
me?
for the panelists. Yep, we can hear you. Go ahead.
Excellent. Yeah, those were awesome talks. And so I'll be moderating the Q&A session, and I'm going to be pulling these questions both from Facebook and from the Q&A function on Zoom. So if you have any questions, please just go ahead and type them in there. We do have a couple questions so far. And this first question, I believe, will be for Dr. Perlman. Can viral RNAs, are they subject to epigenetic modification? And if so, is that, can something be done to sort of help with that process? For example, could you induce RNA methylation with high levels of folate?
Yeah, I don't have a good answer for that. I know that the RNAs are modified. There's N6-methyladenosine modifications. I'm not sure that it means anything other than what it means in most viral infections. So I think this is an area that people actually, now that the SARS-CoV-2 is being so intensively studied, people are going to know more about. Before SARS, the field contained very few scientists, and before SARS-CoV-2, most of those had left the field. So we maintained a reasonable research effort, but we didn't cover anything. I think these kinds of questions are going to be addressed much more in the next few months as more and more people are studying the virus.
Okay, cool. Our next question is for Dr. Harper, and it's so regarding sort of the human ecosystem and how, you know, with the rise in population, we're seeing increased infection rates, but perhaps is there sort of an argument for having different rates of infection, say in urban and rural places? And if we can sort of discern the differences between those two things, is it useful in understanding pandemics?
I think it's definitely an important dimension of epidemiology, and it can be hard for historians to always have the kind of evidence that lets us understand that. But it's clearly one of the really important factors in the way that diseases both emerge and the way that they pose a burden on human societies. And one of the, just to take one kind of example, is the history of urbanization in the early modern period, because in the 17th, 18th century, cities got much, much bigger. And this created an almost entirely novel kind of disease environment where the brunt of the burden was borne by children. And we kind of think of childhood infectious diseases being a sort of universal characteristic of underdeveloped or pre-modern societies. But in fact, really the kind of disease pool that we think of as the set of almost universal childhood diseases, some of which are controlled by vaccines now, some not, really take shape pretty recently in the 17th, 18th century. And I think a lot of I think there's a lot of potential to understand how pathogens evolve, how they specialize in humans, and how they come to be predominantly childhood diseases by understanding more deeply the history and the ecological context of those diseases.
Okay, next question is for Dr. Perlman. Dr. Perlman, what do you think are the biggest molecular factors driving the jump to new species that we keep seeing? Uh-oh, I think you're on mute. Can we unmute Dr. Perlman? There we go.
Now I'm unmuted. Yes, I was.
There we go.
Yeah, now I'm unmuted. Yeah, so the question is, what are the factors that allow for, the molecular factors that allow for crossing species for the zoonotic transmission? Is that basically the question?
Yeah, so basically going from, say, bats to humans and et cetera.
Yeah, so I think it's a really interesting question because coronaviruses seem to hop species so well. And other kinds of viruses do. So there's viruses in Asia, such as one called Nipah virus, which also crosses species, but it really doesn't spread very well from human to human. So the thing that these viruses do, and I don't know if this is related to the huge size of the viral genome or what factors there are, but these viruses seem to be able to bind to receptors, and maybe this is why it uses these big ectopeptidases that are common enough across species. So once the virus crosses a species, it can use the same receptor easily. So that was certainly true for the MERS coronavirus and SARS-CoV-2. With SARS-CoV-1, in fact, it didn't use the receptor that well, and we could watch it evolve over the few months of the epidemic to better adapt to the human receptor. But I don't know if that's the reason or there's another part of the genome that contributes to that. The coronavirus genome really does the same thing that a lot of other viruses do, makes more, it's basically the machinery for making more of the virus. And it also has proteins that help evade the immune response of the host, so the virus can actually establish a foothold. And so whether these are more cross species or not, I don't know. As I mentioned during my talk, a lot of these seem to be able to be eliminated and have the virus still be pathogenic. So I think this is something we just don't really know the answer to yet, but certainly the receptor binding is key. If it couldn't bind to the receptor, it couldn't enter a cell once across species. So that's a very important part of what it does.
Okay, next question for Dr. Harper. I picked up on something you said at the end of your talk about the case study of the Russian virus. And you said that that virus had sort of hung around for about 3 years. Is that something that we can expect perhaps with this virus?
Yeah, who knows? I do think it's interesting, and obviously we'd love to know if it really was influenza, and if it was influenza, what strain was it? But it's worth thinking about the epidemiology of that outbreak, and I think it provides a little more context. And if we want to kind of think comparatively about these big global pandemics of respiratory infections, having the 1957, 58, 1918, 19, expanding that back to the 1889, 92 could be helpful, and just give us a little bit broader basis for trying to think about some of these recent events where you have a globally very interconnected society that experiences these very fast-moving planetary scale pandemics. And what we know about that one, and we know quite a lot about that one, is widely covered in the mass media. So there's a rich kind of basis for understanding it. It moved very, very fast. It was came somewhere out of Central Asia probably, and was in Western Europe within less than a month, and almost simultaneously on the eastern seaboard of the United States. And then within a few more weeks, it reached South America. When it struck, it tended to move very quickly. So it would move through a population in 4, 5, 6 weeks, and then it would recede. And there were these long lulls, so kind of these interepidemic periods locally. And then the next winter, it returned again. So it was very much a winter seasonal pattern. And it came and went for almost everywhere 3 years in a row. So, without knowing what pathogen it was, we can't use that to forecast, but it does just give us another case study to think about, as people are trying to make good assumptions as they build models to think with some of these historical examples. That's another one we know a lot about.
Okay. Next question is for Dr. Perlman. Regarding whether plasma therapy, could that be useful for treating COVID-19?
Yeah, so this is certainly one of the things that people are thinking about now. So the idea is that if you have antibodies to any pathogen and you have it at high enough levels, you can often impact the infection. You can make the amount of the infectious agent decreased. And if you decrease it before it's begun to spread very much within a body, you're going to have less of an infection. So that's the idea here. One of the issues is, who do you give plasma therapy to? So you'd be taking it from people who are recovered from the infection. They have to have a good response, a good antibody response for it to be useful. And one would have to deliver it at early times during the disease course, because at later times it may not help at all. And the other thing is that because convalescent sera or plasma may not be so abundant, it might be more useful doing what we did in Ebola, where we can make specific monoclonal antibodies that target the virus, that maybe using a pool of 2 or 3 of these would be the most effective way to substitute for convalescent plasma. But the key thing is to give it early during the infection. This is not, in Ebola, the amount of virus was always increasing during the infection. In these infections, virus is being eliminated even as people get sick. And in fact, for all these coronaviruses, people don't really get sick until the virus starts being eliminated. So it's so much a part of the host response to the virus that makes people sick and potentially die. So yes, that may be useful, but time will tell how useful it is. And there's also certain people worry a bit about whether there's going to be unwanted responses from mixing virus with plasma. We saw this a little bit in experimental animals, but whether it's significant or not is something we don't know yet.
So to piggyback off of that sort of concept, you mentioned that remdesivir would be most helpful if administered early in the disease. And given what you just said, is it maybe true that administering it later in the disease may not help much, or would it still be beneficial?
Yeah, this is something I would have guessed before all this started that it wouldn't help very much, because if your virus load is already decreasing and helping it go away faster, of course, is good. But if most of your disease now is the host response, if you don't also do something to turn off that host response, then the clinical situation of that patient may not be improved. Obviously, clearing the virus and having the patient die is not an outcome that's acceptable. So we have to figure out how to do both parts. This is one of the problems with remdesivir. Originally, it was used for patients who are on ventilators. Well, that's exactly the group I would have thought it wouldn't help very much. And I don't think there was much evidence it did help those very much.
Okay, next questions for Dr. Harper. Could you correlate written records of livestock die-offs with records of gastrointestinal or respiratory disease in humans? And would this information be useful?
Yeah, I think it's an important question. And we know from historical records that people often observe that livestock die-offs and human diseases coincided. And so one interesting question that raises, to use again the example of measles virus, which is somewhat closely related to the rinderpest virus. We don't actually know as much as we'd like about the emergence of measles virus. It's a highly specialized human pathogen, but at some point it shared an ancestor with rinderpest virus, and we can only speculate what the ancestral pathogen might have looked like and what it might have been capable of, but it could have been capable of infecting both humans and livestock. And so when we read in the historical record about these plagues that affected both humans and animals, it could represent an earlier stage in the emergence of some of these pathogens that we share or that evolved directly from livestock pathogens. I'll say too that there's a lot of evidence, and it's a question that's not well studied, but in the 19th century, a huge number of animal panzootics. So there's giant, well-known rinderpest panzootics that reach Africa with huge consequences. There's a massive equine influenza epidemic in the United States in the late 19th century. That's a very interesting event. And we see in the 19th century where there's also a growth in veterinary medicine, diseases that are probably things like the bovine coronavirus is spreading. So bovine pneumonias that are spreading very rapidly in the 19th century. So with the industrialization of the economy and the rise of kind of mass scale livestock farming in the 19th and 20th century, it's undoubtedly been a kind of stew where pathogens have evolved. And I think we can trace the evolution and emergence of some of these animal pathogens that Stanley started talking about, at least loosely in the historical records in the 19th and 20th centuries.
Okay, I think we have time for one more question. This would be for Dr. Perlman, and it's very specific. Okay, in a research paper, it was found that RBD of SARS-CoV-2 remains, or SARS-CoV-2 remains hidden to evade immune response and only stands when ACE2 receptor is targeted, what might be the signal for RBD to stand to bind to ACE2? Uh-oh, you're muted. There we go. Okay.
Yeah. So when you say, what does the questionnaire mean by stand? Do you know? Maybe
presentation.
So this is a technical question. There's a technical.
Yeah, it's very technical.
Okay, so because it's an interest. I think the person who's asking the question is asking about a very recent and unique phenomena, which is that when I was discussing the S protein of the SARS and MERS coronaviruses, I talked how the part of the surface protein that binds to the receptor is either in an up position or a down position. And it turns out for SARS-CoV-2, it always is in a down position, and it needs to be in the up position to bind to the receptor. Okay. So, and that may be what the question is.
I think that's what it is, yeah.
Yeah, so it's really interesting phenomena because we know that that the SARS-CoV-2 S protein actually binds better to ACE2 than SARS-CoV-1 does, or we think the NL63 one, which has the same receptor, but we don't know for sure about that one. Yet it seems to be in this down position. So what we think is that, in fact, there probably is a preceding event that we just don't understand yet that people are investigating that allow that ACE2 or the spike protein to put it in an anthropomorphic term, feel more comfortable with being in an up position so then it can bind to the ACE2 receptor. But again, we don't have much information about that preceding event, but that's what you might need something, because if you have a protein that's always hidden, it's not likely to be able to bind to its receptor. So this is really an interesting area of investigation.
Okay, I think that's the questions. Yep, I'll turn it back over.
Okay, I was just going to say we probably need to wrap up the webinar. And I want to first of all thank both of our speakers for great presentations and question and answers and so on. I think it's going to do it for the first SEBM webinar. It's, we've set the bar very high, and thank you very much. I look forward to invite people to also join us next week, next Wednesday, May 20th, where Dr. Stephen Goodman will lead a second pass of the COVID-19 webinar series with Dr. Colleen Johnson, who will talk about clinical trials and what tools do we have now and how can we use them type thing. Again, I want to thank the sponsor, SEBM, for sponsoring the webinar. And if you have an inkling to join SEBM, please contact us through our executive director, Jessica Homa, on our website and so forth. And we'd be happy to entertain folks joining and continuing with our concepts and activities. With that, I guess we call it to an end, and thank everybody for participation, and look forward to next week.
Thank you all.
Thank you.